Hello friends, how are you? My name is Rizwan Hafeez, and in the previous video we were talking about the hallmarks of cancer. We have also talked about the genetic aspects of all the hallmarks of cancer as well. In this video I will be talking about proto-oncogenes and oncogenes. Well, more specifically I'll be talking about the proto-oncogenes. You know that the mutated form of proto-oncogenes are basically called as oncogenes. So I will talk about the proto-oncogenes. I will try to elaborate this concept with examples. Okay, in the previous video I told you that there are some genes which are required for the DNA replication. Okay, and these genes will be stimulated, will be expressed in G1 phase, and when the product of these genes will be formed in G1 phase, then in S phase DNA replication will start. In S phase DNA replication will start. So for example, here are those genes. The expression of these genes will help in the DNA replication. And if there is a DNA replication, so we can say that these genes are basically going to help the cell proliferation, cell division. Obviously, in the previous video I have talked about that in G1 all those enzymes will be synthesized which are required for the DNA replication, and DNA replication will take place in S phase. In G2 phase all those enzymes will be formed which are required for the mitosis, for the division of the cell, for the proliferation of the cell, and in M phase division of cell will take place. Then some of the cells will go into the resting state called as G0 until the next signal for the replication or cell division come, and some of the cells will keep on dividing. Okay, so for example, these are the genes which are responsible for the DNA replication. Okay, I have also talked about that there are some other set of genes who will stimulate these genes in a positive sense. They will stimulate these genes, and those genes which stimulate these genes, obviously they are basically helping in the proliferation of the cell. And those genes who are stimulating these set of genes are called as Proto-Oncogenes. These genes are not called as Proto-Oncogenes. These are the genes which are required for the DNA replication. And that all other genes, the product of those genes will stimulate these genes. Those genes will be called as Proto-Oncogenes. Okay. Similarly, at the same time, I also explained about that there is another set of genes who will be inhibiting after certain proliferation and cell division. Now we need to stop further cell division and further proliferation. And these genes will play their role at this time, and they will give inhibitory signal to these genes so that the expression of these genes, the replication and translation of these genes will get stopped. And those genes who are giving negative signal, inhibitory signal to these set of genes, those genes are called as tumor suppressor genes. Tumor suppressor gene. The genes who are stimulating this set of genes are called as proto-oncogenes, and the genes who are inhibiting this set of genes are called as tumor suppressor genes. In today's video, I will be focusing on the tumor suppressor, proto-oncogenes. In the upcoming video, I will talk about the tumor suppressor genes. Clear? So let's talk about the tumor suppressor gene. And the first gene, this tumor suppressor gene is basically a set of genes. There are lots of genes who are basically helping a cell to proliferate, to replicate more and more, to divide more and more. So the set of genes who are basically helping a cell in proliferation are called as proto-oncogenes. Okay, and if there is a mutation in those genes, if there is a mutation, obviously they will start giving more signals for the proliferation and for the replication. And if there is a mutation, is in a sense that the function of these genes increase. Like gain of function mutation happen in these genes, it will result into a cancer. Obviously they will stimulate more and more these genes, and the proliferation and the cell division and the DNA replication will increase, and there will be more and more cell division. And you know that if there is uncontrolled cell division, that will be called as cancer. And it's a genetic disorder. Obviously, if there is a mutation in any of the genes who belong to Proto-Oncogenes, that may lead to the cancer. Okay. Now, in the next video, I will talk about the tumor suppressor gene. The first genes which I am going to talk about that is related to the Proto-Oncogene is growth factor genes. Growth factors. Obviously, when these genes will be stimulated, RNA will be formed. Then RNA will go out of the nucleus. It will bind on the ribosome. Protein will be formed, and that protein will be a growth hormone. A growth hormone. A growth factor. Okay. And now you know that growth factor will go out of the cell. Will go out of the cell. Similarly, the next genes, if there is a mutation in the growth hormone genes, the more and more growth hormone will be formed. And obviously, if the synthesis of growth hormone has been increased, obviously it may result into higher proliferation, and that may lead to the cancer. So this is the reason I am talking about that if there is a mutation, if there is a gain of function mutation in the Proto-Oncogene, not loss of function mutation. If there is a gain of function mutation in the Proto-Oncogenes, this type of mutation may lead to the cancer. Clear? Okay. Now the other genes which I am going to talk about is growth hormone receptors. And obviously when these genes will be expressed after transcription, after translation, when a protein will be formed, that protein will be a growth hormone receptor. And growth hormone receptor are supposed to be attached on the cell membrane. After maturation, these protein will come and they will attach on the lipid bilayer membrane of the cell. Clear? Now, third protein, at the end I will link all these protein. Okay? The third protein which I am going to talk about is a transducer protein. Transducer, who is responsible for taking message. Who is responsible for taking message from the growth hormone receptor to the DNA and to the other transcription factor and to the other proteins as well. And one of the transducer protein is RAS protein. RAS protein. Okay, and the gene of this RAS protein is also basically Proto-Oncogene. I will explain. I will link all these things. I will link growth hormone with the receptor and with the RAS gene and with the MYC. Just give me 1 minute. Let me first of all let me tell you what are these genes. Now the fourth gene is basically called as MYC gene. MYC gene, and the product of this gene will be MYC protein. MYC protein, and MYC protein is a global transcription factor. It's a global transcription factor. It can activate lots of pro-proliferative genes. It can activate those genes which help in the proliferation of cell, which help in the cell division. So this MYC is basically a transcription factor. It's the product of this gene, that is MYC protein. That protein is basically a transcription factor, and that transcription factor can activate lots of other genes, only pro-proliferative genes, which can stimulate the process of proliferation of a cell, which can stimulate the process of cell division of a cell. Okay, and approximately 50% of the total genome, 50% of the total genes of our body are stimulated by this transcription factor, that is MYC. And this is very important. If there is a mutation in this MYC, it can lead to lots of many types of cancers. Approximately 40% of the cancer are happened because of mutation in this MYC gene. And MYC gene is a Proto-Oncogene. So if there is a mutation in MYC gene, obviously to develop a cancer, there should be gain of function mutation. So if the function of MYC will increase, if the function of MYC genes will increase, obviously what will happen? It will stimulate more and more. It will stimulate pro-proliferative genes more and more, and the process of proliferation will increase, and that may lead to the cancer. Clear? Okay, now let's link all these things. So what will happen when this growth hormone, it will bind on its receptor. It will bind on its growth hormone receptor, and this RAS protein, that is the product of this RAS gene, this RAS protein is attached with this receptor, initially this RAS protein is not activated. Why it is not activated? Because it is bound with GDP, guanosine diphosphate. It will be activated only when GDP will be displaced and GTP will be attached. Now please listen to me carefully. Growth hormone will come, it will bind to its receptor, and when it will bind to its receptor, there will be conformational change in the receptor, and this conformational change will cause a conformational change in this RAS protein as well. And because of these conformational change, the GDP will detach and GTP, let me make 3-phosphate, okay, GTP will attach over the RAS protein. And when the GTP will be attached, GTP will be displaced, RAS protein will get activated. RAS protein will get activated. And now this RAS protein will send a message through a signal, 2 pathways. The one is MAP kinase pathway and second one is AKT pathway. Because of so through these 2 pathways, this RAS protein is going to activate MYC protein live. It is going to activate the MYC protein. MYC protein is a basically product of MYC gene. Okay, and now this RAS protein will activate MYC protein further. It will increase the lifespan of MYC protein. How? By phosphorylation, by attaching a phosphate group PO4-3. By attaching a phosphate group over the MYC protein, MYC transcription factor, it will activate the MYC protein and it will increase the life of MYC protein. By this pathway, that is called as MAP kinase pathway, it will phosphorylate serine amino acid present at the 62 number. It will attach this phosphate group on the serine amino acid. You know, this is a protein and it is made up of lots of amino acid. At the 62 number, the amino acid that is present is called as serine. So by this pathway, the RAS protein will attach phosphate group on the serine 62. It will cause phosphorylation. And by AKT pathway, it will attach this phosphate group on the threonine 58 amino acid. At the end, ultimately, cutting the long story short, this RAS protein is basically activating this MYC protein by phosphorylation, by attaching phosphate group. And once this MYC will be activated, it will activate lots of other pro-proliferative genes, and the process of replication and process of cell proliferation will start. So it is happening in a normal mechanism. But if there is a mutation in this growth hormone growth hormone genes, so more and more growth hormone will be formed. It will keep on forming, keep on forming, keep on forming. And now this signal will increase and the phosphorylation of MYC will increase. And what will happen? The proliferation of cell will increase. Cell division, cell replication will increase. And that may lead to the cancer. That may lead to cancer. Similarly, if there is a mutation in gain of function mutation, not loss of function mutation, please remember it. If there is a gain of function mutation in growth hormone receptor, now there will be lots of growth hormone receptor will be present over the cell membrane of a cell. And the more and more signal will go. And MYC life and its activation will increase. And obviously, the process of cell proliferation, replication and division will increase. And that may lead to the cancer. If there is a mutation in the RAS protein, obviously RAS protein will keep on sending the signal without being activated by growth hormone and growth hormone receptor. It will send signals spontaneously without being activated by the RAS protein receptor. And obviously it will overactivate the MYC transcription factor, MYC protein, and that may lead to the cancer. So if there is a gain of function mutation in growth hormone receptor, RAS protein, or if there is a mutation in MYC protein, growth hormone is normal, growth hormone receptor is normal, RAS protein is normal, but MYC gene has been overactivated. Why? Just because of mutation. Just because of gain of function mutation. So obviously it will stay more active. It will produce more. It will be produced more and more. It will be expressed more and more. So what will happen? Obviously the process of replication and proliferation will increase. Getting it? Now what will happen? Let me proceed forward. This MYC gene, I have explained that it can activate lots of other pro-proliferative genes. Similarly, it is going to activate two very important genes. The one of the gene, the product of this gene is cyclin. It will also activate cyclin because it is a transcription factor. So it will help in the transcription of this gene. And after transcription and translation, the product will be cyclin. It can also activate another gene, and the product of that gene will be kinase. But this kinase is very unique. Why? Because it depends upon the cyclin for its proper function. So that is the reason this kinase is basically called as cyclin dependent kinase, CDK. So MYC will activate cyclin, and MYC transcription factor that will also activate or produce cyclin dependent kinase, CDK. Okay. Now, what are the function of cyclin and CDK? We will talk about 1 or 2 minutes. Let's start another story. Let's talk about another story, but that is very important. So here is a set of genes, and if these genes will be activated, the replication of DNA will increase. I have explained about it. The replication of DNA will increase. Getting it? Okay. Now what will happen? There is a transcription factor, E2F. E2F is basically family of transcription factor that activate lots of pro-proliferative genes. And one of the set of genes is this set of genes that belong to the that is responsible for the cell replication. And these genes will be activated by this E2F transcription factor. E2F transcription factor. But this E2F transcription factor has been bound, has been arrested by this RB protein, retinoblastoma protein. This RB protein is not letting E2F to transcribe these set of genes. But these RB protein, it will allow these transcription factor for the transcription once this RB protein will get phosphorylated. Again, I will repeat. These are transcription set of genes responsible for the DNA replication, and these are the transcription factor for these genes. But these transcription factor, they are not free. They are attached with the RB protein. So this is the reason they are not functioning properly. They are not helping these genes to get transcribed. Getting it? Now what will happen? Once this RB protein will get phosphorylated, it will allow these transcription factor to go and start the process of transcription for these genes. And now these CDK and cyclin, they are basically responsible for the phosphorylation of these RB protein, retinoblastoma protein. Now when this cyclin dependent kinase will cause the phosphorylation of retinoblastoma protein, now what will happen? This RB protein will allow E2F to go and start the process of transcription of these specific genes that are responsible for the replication of DNA. So basically what will happen? These CDK are basically increasing the process of replication, process of proliferation. So they are also they also belong to a Proto-Oncogenes. Not this RB. It was basically inhibiting the replication process. So this RB gene will be a tumor suppressor gene, and we will talk about it in the next lecture. Okay, I think you are very much clear about the tumor suppressor, Proto-Onco gene. So if there is a mutation in CDK gene, gain of function mutation, so what will happen? It will cause phosphorylation of these RB protein. It will cause more and more and more phosphorylation of these RB proteins. And if the phosphorylation of this RB protein will increase, obviously the process of transcription of these genes that are responsible for the DNA replication will increase, and the process of proliferation and replication will increase. Process of proliferation and replication will increase. Similarly, if there is a mutation in cyclin, again the process of replication and proliferation will increase. So if there is a mutation in any one of these genes that belongs to a group of Proto-Oncogenes, if there is a gain of function mutation in any one of the genes, that may result in cancer. So this was a brief introduction about the Proto-Oncogenes. In the next video, I'll be talking about the Tumor Suppressor gene. I hope you like my video. Please don't forget to subscribe my channel and press the bell icon so that you may get notification for the future videos. Thank you so much.